Welcome, everyone, to another episode of the Pazio Institute Interview Series. Today, we have with us distinguished guest Ryan Smith. He is the founder of TruDiagnostics. It's a leading CLIA-certified lab specializing in epigenetic testing and insights into biological aging. Since launching in 2020, TruDiagnostic has built the largest private epigenetic database in the world, testing over 130,000 patients. Ryan's team has created innovative algorithms to predict inflammatory markers, disease diagnosis, telomere length, aging pace, immune cell subsets, cellular replication, and much more. They're actively involved in over 85 clinical trials, investigating the best ways to reverse these markers through diet, lifestyle, and medical interventions. On top of that, Ryan was involved in peptides, and that's how I met Ryan. Was through peptides, and we worked for a long time together, and he founded that group that was going into all that, and that pharmacy still exists. But with all the troubles that we're having with peptides right now, where I'm so glad that Ryan's at TruDiagnostic, and really, honestly, he's pushing the envelope on these tests. And we're going to dive deep into what is epigenetics, why do we need to know it, is this data going to be something that's going to give us feedback so that we can change your protocols so you can live a longer, healthier life without having single point failures? And how does this relate to medical maintenance engineering that we're using here at the Pazio Institute and at the Institute for Medical Maintenance Engineering? So that said, welcome, Ryan.
Yeah, thanks so much for having me. It's good to catch up again. I know we've obviously worked together for years, and happy to be on.
Awesome. So let's give people a little bit of background, right? If you go and you're looking in your AI and you type in epigenetics, or you type in, what is my biological age versus my chronological age, you really dive deep, you're going to see there's a ton of clocks out there. Right. And a lot of these clocks, are suspect. Some of them have some pretty good data behind them. So we're going to dive deep into it and, teach you about what is epigenetics, what is methylation, what are these CPG markers, and, explaining then, and why do we pick certain markers? What is the evidence behind all that? And why do we want to pick these specific markers, and why would you not want to pick other markers?
Yeah, absolutely. I always like to start with like the background on aging. I think it's always, the easiest way to go because, as you mentioned, it can be overwhelming to figure out, what's the best way to measure my aging process.
Yeah.
And, even since the 1960s, people have been doing really, really crude methods of biological aging, right? Like, your chronological age plus one year for every pack per day you smoked. And over time, these have gotten much, much more sophisticated to a point where now we can actually use them and take them seriously, where we can actually, sort of quantify this process in real time. And obviously, I don't want to understate the importance of that because I want everyone to know that aging is the biggest risk factor for every chronic disease and death. And so, as we're talking about, the one thing we really want to measure, no one would, with a history of cardiovascular disease in their family is going to ignore the biggest cardiovascular risk markers, right? No one who's worried about Alzheimer's disease is going to, forget about the biggest risk factors there. But if everyone's healthy, and really not diseased, and we don't have to worry about a particular disease process, this aging measurement is one of the best things that everyone can do.
Awesome. Yeah. And, just so everyone knows, if you're not a member here at Pazio Institute, we're running multiple of these tests. TruDiagnostic being, one of the main tests that we do. But there's a lot of different ways that we can measure aging. It's just that epigenetics has shown to be very useful, and that it's responsive to these therapies. Ryan shared with me a paper a while back that came out at the end of last year and showed the efficacy of different interventions on these tests that will show that you can lower the rate of your aging. And now some of these tests, you can actually be organ specific.
Yeah. Yeah, it's come a long way. It used to be that we could get an approximation, of how you're aging. We couldn't tell you why. Now we have individual resolution where we can sort of not just tell you if you're faster or slower, but actually what's driving that process. And that makes it actionable, right? So that we can actually, target specific interventions, focus on individual things. And so this whole field is really exploding. I mean, these first clocks that came out with epigenetics really started in 2013. So we're over a decade away from that now. And at first they were used to just tell someone's chronological age, right? Like if they left DNA at a crime scene, or
Right.
if they were dating refugees to see if they were adults or minors. And then, but over the years, these have gotten progressively better to provide that resolution, as you mentioned.
Great. Yeah. And, Steve Horvath is pretty famous for, one of these first clocks, but now these next clocks, they're using way more methylation markers. And now we have machine learning and we have AI that's diving deep into these markers, and that, we can identify which of these markers make more sense to us and which ones don't. Talk a little bit about like where it started and how are the clocks getting progressively better and why?
Yeah, yeah, definitely. So to start in 2013, as I mentioned, at first it was meant to be just a chronological age predictor. That's sort of the method that was being used. But the problem is we don't really want to know your chronological age, right? It's sort of a relatively
Yeah.
arbitrary number. It's the number of trips you've taken around the sun. It doesn't necessarily inform us of what's happening in your body. And that's why,
Right.
we can see this in people who, are maybe in their 70s who look like they're 50, and then vice versa. This is something we come across all the time, but we need a way to molecularly measure something that's, completely independent, not influenced by, anything other than data, right? And outcomes. And so this really started in 2013 at UCLA with Steve Horvath. And again, first trained to predict the chronological age of an individual. And it wasn't really used as a medical tool until they used it in really large populations. And they saw that those with younger biological ages than their chronological age were protected from negative outcomes. And those who were older with this testing than their chronological age were more at risk for negative outcomes. So that's why it was so exciting, is that all of a sudden we had a molecular measure that could predict outcomes. And ultimately, that's what we want, because unfortunately, with aging, if we really want to have an impact, we can't wait 40 or 30 years to figure out what happens to people as they get older. We really need something that can change immediately so that we can figure out what's working on an individual basis, but also what's working just generally. And so these biological age clocks have been used as sort of surrogates for prediction. And so at first, what we call these first generation clocks, those are trained at chronological age, and those are largely not very important anymore. They're super accurate at predicting chronological age. Our mean absolute error now is around, I think, 5 months. So we can tell you within a
Wow.
5-month window exactly, your chronological age. But again, that's not what we care about. So in right around 2017, Steve Horvath's postdoc, Morgan Levine, who's now gone on to be one of the most important people within aging, she created the first second generation clock. And these are trained not just on your chronological age, but on biological phenotypes. So things like your blood-based biomarkers, for instance. And that took a big step forward, because now we're training it on data, health data, that's really important to us. Just like if you go to any physician, you'd get your bloods done. This was sort of a surrogate to predict aging through that method, and it was a big step forward. And so that's one of the biggest things. And then now we even have things like third generation clocks, which are tracked in the same patients longitudinally, so we can actually see on like an individual basis how things are going. That's that pace of aging clock. But ultimately, the one thing that's making these clocks really, really good and a lot better is the AI methods to train, but also the data used to train. Which is now, the original clock by Morgan Levine had 9 blood-based biomarkers. Our most recent collaboration we did with Yale to create Symphony Age, which gives you the age of each organ system, had over 300 different biomarkers that was used on every single patient. And in our Omic age, we measured over 10,000 biomarkers used in every
Wow.
single patient. And so the data, to get a full picture of everything that's happening in your body, we're really trying to get as much data as possible. And the more data we get, the better we can get at predicting outcomes, which ultimately is what we care about when we're creating these clocks.
That's fantastic. I'm going to back up a second just so you guys
Yeah.
understand. When you have your genome, and that we can't change your genome. Well, we're going to be able to change your genome. Liz Parrish, she's on our advisory board, and her genetic changing, and that's coming, right? So we're going to be able to do some changes there, and that's going to be next game. But what I want to focus on is you have your genome, but then you have your phenotype. And it's like how your genes are being expressed. And a lot of that has to do with your epigenetics. And some of that's with your family tree and what's happened to them and what's happened to you over your lifetime. So one of the things that we're really focused on is, all right, if you hypermethylate something, right, that's you're going to suppress that. And so most of these sites are these sites that promote a gene to be expressed. So think about that as a program that needs to be run. And if I hypermethylate something, right, and I suppress it, and that gene is what's called a tumor suppressor gene, now you're at higher risk for cancer, right? Or if we hypomethylate things, now we can destabilize that gene. And so we don't really want all that. And we want to know what's happening because a lot of changes in methylation change inflammation. We know inflammaging is a huge part of aging, and we want to turn that down. It changes your metabolism, and it also can change your DNA repair enzymes, which has some really negative consequence. So these are some really good things we want to know. And now the next generation stuff that TruDiagnostics is doing can even dial into like your organs and stuff. And so, I really want to dive in, Ryan, to, what you guys are doing with Symphony Age, with Omic aging, how we do that. And let's give people a little bit of background on omics in general.
Yeah.
This word gets thrown around a lot. And I don't think people really understand what omics are. And there's so many different types of omics. Like we have phenomics, we have metabolomics, we have proteomics, we have transcriptomics, we have
Yeah.
epigenomics. And so, why do we want to know all these different things? What are they going to tell us? And then what can we do about it?
Yeah, this is omics is one of those things that is just now also exploding because
Yeah.
we're getting so much data, and now we have the ability to interpret that data with AI. And so now, it's sort of this race between the data we can generate and the AI to interpret, right? And so
Right.
to start with, to omics, omics is just sort of a classification of what we can measure within the body. It's sort of like a framework for what types of things we can actually test. It's very unidirectional. So it starts first and foremost with genomics, right? This is the basic building blocks of our DNA. This is where we start. But then we go to epigenetics, and epigenetics is what turns things on or off, right? As epi stands for above the genome, and as you mentioned, methylation is the off switch there, but there are a couple on switches too that we can measure. But the important thing here is that as we first start out as a pluripotent stem cell, we've got to create all of these different cells in our body. And so the way that I like to always reference it is to say that your skin and your brain and your heart all have very different roles, right? They all have very specialized skill sets, but they get those by the instructions epigenetically, right? They all have the same DNA, but what's turned on or turned off gives the cell the identity to do its specialized behavior. And so we're measuring DNA methylation particularly there, as one of our main outputs. So with that being said, though, then DNA methylation leads to what genes are on and off, and that creates RNA, or transcriptomics, as they would call it. And those RNA and go to ribosomes to create peptides and proteins. Those peptides and proteins are called the proteomics, and then they really create the infrastructure of the body, right? They sort of create the all the things that sort of manifest, and then ultimately they have processes. So it might be, for instance, creating hormones or breaking down hormones, and as we start to do things like that, or we have exposure to chemicals in the environment, or for our diet nutrition, that's the metabolites. So the breakdown of those different processes that happen, and then ultimately we have phenomics, or what actually happens to us. Do we have gray hair, blonde hair? Do we have are we quick and fast at running, or are we a little bit slower? But all of those things are the ways we classify the measure. And so that's the sort of the idea of omics, just sort of how we're sort of categorizing all the things that we can measure in your body to give us information about your health.
Awesome. So that said, let's dive right into Omic Age then. How
Yeah,
you developed Omic Age and how we can use it as clinicians and in medical
yeah.
engineering to tailor that person's age reversal protocol and which pharmaceuticals we're going to use and lifestyle interventions.
Yeah, definitely. So as I mentioned, as the clocks have progressed, they've gotten better, and the way they've gotten better is by adding more data. And so we had one, pretty big idea when we first started, which is we wanted to create the best in class epigenetic clock. And in order to do that, we needed as much data as possible. So in collaboration with some Jessica Lasky-Sue at Harvard and her lab, we built a huge data set within the Mass General Biobank. So in 5,000 people, we measured all of those omics that we talked about: full genome sequencing, epigenetics, transcriptomics, metabolomics. We measured over 3,500 metabolites. We measured over 8,000 proteins. We measured over 70 clinical values, in addition to a million different methylation locations and the full genome sequencing. So it's a lot of data. If we were to really do this on every single person, it would cost $10,000 to $15,000 per person, which is really exciting because we get a lot of data, but it's really not scalable, right? That's not something that most people can afford, and it's not something that is even the most actionable even right now, because it's so much data, it's really hard to interpret. So what we decided to do with Omic Age was to see if we could just use DNA methylation to predict some of the other omics. So could we, with just DNA methylation, predict your cholesterol levels, or could we predict your inflammatory levels, or could we predict your diet behaviors even? And the answer was yes, we could, and actually much better than we anticipated.
Yeah.
This had been done before with even some other clocks where they tried to predict 8 plasma proteins. The only difference is we did it in a much larger scale. And so instead of just getting 8 or plasma proteins that we're predicting, we got 1,700 variables that we could predict just with DNA methylation. And so it sounds a little bit like Theranos. That's why I don't come to these interviews with my black turtleneck and raspy voice. But it's actually really cool science right now, is that with just a couple drops of blood, we can tell you a lot of resolution. We can tell you, for instance, what environmental chemicals you've been exposed to, how much have you smoked across in your entire lifetime, how much are you drinking. We can tell you about if you're taking aspirin or ibuprofen or acetaminophen, right? We can sort of break all those things down based on now just the expression of your DNA, which is pretty incredible. But in the case of Omic Age and aging, what it does is it gives us resolution into the why you're aging. So, for instance, some people might have, I already have mentioned things like inflammaging. People might have really high inflammation. That's leading to their aging. Other people might have a lot of toxic exposures, like PFAS chemicals, for instance.
Mhm.
Some people might have NAD deficiencies, as we've talked about. And so there's a lot of reasons why people can age. But now with these what we call epigenetic biomarker proxies, or EBPs, that we included into this Omic Age, we can give you a score of how old you are, but we can also tell you what is out of range, what's higher than it should be, or what's lower than it should be. And that's super important to giving, again, really good and personalized interventions.
That's fantastic. All right. Let's do your symphony age now, and then we can
Yeah,
talk about some of like the rate of aging and some of
yeah,
the clocks that that you're in, like grim age and things like that. Um,
yeah.
we can tie it all together into how we're gonna treat people.
Yeah, certainly. And so one of the things that people get frustrated on, I think a lot of times when we talk about clocks, is that there are so many of them, right? I think we just talked about the four that we like the most, which is Grim
Yeah.
Age, Symphony Age, Omic Age, and Dunedin Pace. But with that being said, even if we're talking about the good clocks and we have those four, the question is, why are there so many, right? And the answer is that the definition of what aging is still a little bit controversial. In the case of Grim Age and Omic Age, we trained these algorithms to predict time until death. So they're some of the most accurate predictors of death available. I mean, in case of Omic Age, we can predict death with a 92% accuracy rate within a 5-year time period, which is pretty incredible. That's you're certainly really accurate and better than anything else that's out there. Same with Grim Age, train until time until death. But oftentimes that's not all the things that people would define aging as. Some people like to define aging as health span, right? Like the number of years you're living without a disease or something that's limiting your body. And generally things like Dunedin Pace, which measures sort of the pace of aging across time, is more correlated or predictive of outcomes like mental processing speeds, IQ, facial aging, the ability to move and stand about the world. And so all of these algorithms are trained off of slightly different definitions of this aging. And so Symphony Age is trained on an organ-specific basis of aging, where it takes blood-based biomarkers of each organ system. So, for instance, if we're looking at the brain, we might do things like mental recall tests, or we might do homocysteine, or we might do inflammatory markers. If we're looking at the liver, we might do liver enzyme testing. We might do albumin, some of those other things. And so Symphony Age is one of those that also provides resolution, so we can sort of say what organ system is aging the quickest or the slowest, and which one do we really need to focus on. But each of those have been grouped slightly differently to give us basically a score of aging across 11 different organ systems.
That's fantastic. And like what I really like about Symphony Age and just, Omic Age in general is that we're already getting like full body MRIs. We get volumetric MRI of the brain, so we can, quantitatively look at the different structures and the sizes of the brain and if there's asymmetry. And then we get all the lab tests. And so we're taking all this and we're putting it side by side and using our data lake and our own AI to start looking at this stuff. So, and what's nice is that it's correlating with what we're seeing in those tests. And for someone's cognitive abilities, we use a test called the CNS Vital Signs, which is really an inexpensive test, and it gives you great data on how you're performing. What's your memory like? What's your reaction time like? All this kind of stuff. And then, we have all the other data to put it with. And the nice thing about the epigenetic testing is this is how well your biology is reacting to this and how fast you're aging. Because if I get a CNS vital signs test now and then a year, I just get to see the difference. But why, right? And then once we know why, it'd be like, well, you say your diet's good, but probably it's not.
Yeah, exactly.
And then we can help people change. And the one thing that we find that's really difficult here at the clinic, and why we have patient liaisons checking in with people at regular intervals, is that behavior is the hardest thing to change.
Yeah, you're absolutely right. I mean, we're all locked into our traditions, right? And comfort, sometimes to our detriment. But that's one of the things about epigenetics, is that it's influenced by everything that we do, right? It's influenced by diet and lifestyle. It's influenced by how much we work out, our medications, but also even things like close relationships, right? Like, how happy are we can affect it. And, there are really cool anecdotes here that I always like to throw out just to get people excited about all the things that epigenetics can do. But one of the interesting things is we can still see epigenetic signals of people who have had family in the Holocaust, which has been passed down over time, or the Rwandan genocide. We can predict what zip code you live in based on pollution markers within your DNA. And even in the animal or plant systems, one of my favorite anecdotes is that broccoli is now epigenetically expressing as cauliflower due to climate change. And one of my favorite other sort of species related anecdotes about epigenetics is that if a queen bee dies, the hive has to replace the queen bee, but the queen generally lives 10 times longer than everyone else in the hive. So whenever they have to replace this queen, they use another sort of bee from the hive, and that bee then automatically epigenetically adjusts and lives 10 times longer than what it was previously. And so the epigenetic regulation we know has ties to longevity, but the bigger thing is that almost everything we do plays a role. And if we can read those signals, we can get a really objective feedback on the things that we're doing and how it's affecting us.
That's awesome. And it's just another like reminder to everybody like that science is moving us forward. But right now, your lifestyle is the most important of all of the variables. And we use rapamycin, we use dasatinib, we use supplements, we work on your NAD, we detoxify you, we work on your hormones. All of these things are amazing, but if you don't get your stress under control and you don't get your lifestyle under control and you don't sleep well, you don't eat well, you don't exercise, like you're not going to do that great. And and that's why we really want to promote how easy it is to do things here. So like we prepackage your supplements. And we have ways of tracking your compliance, and it's not just like we're trying to grade you, we're trying to help you. And we're not trying to give you like 50 bottles to take every day. We're trying to make it as simple as possible and follow up with all these things. There's a really interesting epigenetic study that when I first got into epigenetics, I found out about, and it was a mouse in a maze. And you know this study, and they had this female mouse, and she would run around the maze, and they put a little red fire hydrant in this maze. And when they put the little red fire hydrant and she could see it, they gave her a little electric shock. And then they kept moving the fire hydrant around, and every time she saw it, they'd shock her, but they didn't shock her that many times. And then when she saw it, she would just jump, even though she didn't get shocked. And then they took the red fire hydrant out. She had 7 generations pass. They take the 7th generation mouse, they put it in the maze. The 6 generations never saw the red fire hydrant before. She sees the red fire hydrant and she jumps.
Mhm.
And
Yeah, it's incredible.
it's
Yeah.
just incredible how this data, this memory is encoded into our epigenetics. And
Yeah.
I think it's just a good reminder to people, like if they're like, well, I'm just anxious.
Mhm.
Well, maybe not. Maybe one of your ancestors was anxious.
Yeah.
Right?
It's incredible. Yeah. There's other studies like that as well. I mean, even we can see people from even epigenetic signals from the Irish potato famine. Like it's incredible. We talked about DNA as the hardware and epigenetics as the software, and we've accumulated these software features, throughout generations, and it's good because it provides another way for us to adapt, right? And to have optimal survival. So it's just another mechanism, but it's one that's super important and just now beginning to become, I would say, really, really integrated into scientific development.
Great. And I think this is a really great time to start talking about the paper that you shared with me. So if you're looking for this, guys, just look at it's DNA little m, right, which is methylation of DMM. Aging markers are responsive, and it's insight from 51 longevity interventional studies in humans. It's a fantastic paper. I dove into this paper in detail, and overall, what it's saying is that if you change your lifestyle and you do these interventions, especially if you add in age reversal techniques like we're using in medical maintenance engineering, that you can really change your biological age, and you can change it for the better, and you can reduce disease outcomes and increase your health span as well as increase your lifespan.
Yeah, definitely. And that's an important point I want to mention as well, is that it's not just about how long you're living, right? And I think that sometimes people get turned off when we talk about longevity in that fashion, because they think about, living until they're 105, maybe a poor quality of life, or someone having to take care of them. And they're not mutually exclusive. As we're improving lifespan, we're also improving health span to keep you, living the life you want to live longer. But yeah, I think that this paper was exciting because we were able to finally really grade a lot of interventions with all of the different tools to figure out, first off, what was the best tool, but also what interventions were working. And one of the other things I like to say here is that one of the original statistics that Morgan Levine shared with PhenoAge is that if everyone in the world were to reverse their epigenetic age by 7 years, we would cut the incidence of disease in half. 50% of people would no longer be getting sick. And so again, treating aging as a primary outcome has massive benefits to everyone. And if we all want to avoid disease, it is something we can pay attention to. And now we have the ability to measure it, but also to affect it. And I think that's the most exciting thing about all of these different trials. And so we did this with Yale also, and we contributed probably about 15 to 20 of those different interventional trials. But now we've even got over 100 that we'll be doing in some additional follow-ups. And we had some things that were really surprising, but we also had a lot of things that made sense.
That's incredible.
Yeah.
So, and when you guys get your tests back and I go over all these things with you, it's not a report card, right? We're trying to show you what you can do to help yourself. And to Ryan's point about health span, right now, the average person in the United States, the last 10 years of their life is spent in some type of decrepit state. Now, that's either like a dementia, or you're in a wheelchair, or you can't do your daily, acts of living. You can't tie your shoes, you can't cook for yourself, stuff like this, and you're dependent on somebody or a facility or things like that, and you don't have all your faculties. So what we're finding is that that 10 years can be compressed to less than one if you're following a really good lifestyle and you're getting these tests done. And that buys you time, because even if you didn't extend your lifespan, you're extending your health span. And so those years are going to be quality near the end of your life. My grandmother used to always say this saying in Greek, and it was basically she would pray to God to take her like a little bird. And I used to think it was so morbid, but like I totally get it now. And like the bird sitting there, tweet, 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 and then it just falls over dead. And ironically, that's exactly how she went. um,
Yeah,
if we could all just die like that, that would be great, without suffering and, being
yeah.
in hospice or being in a nursing home or things like that. And
Mhm.
we can extend life, even a small percentage, like you were saying, 7%. And, I think that
Yeah.
some of the data is showing we can even do better than that.
Mhm.
And if you start at a decent age, not like when you're 80 at some of this stuff, and say you're 45 years old, and your family history shows that most of the people in your family live to be 90. So you got about 45 years left, and we can add 10% onto that, and then we can decrease your decrepitness. You bought yourself more than a decade of life, probably 15 years extra that you wouldn't have had to do things. What's that worth? And that's with
Yeah.
technology. What about the technology in 20 years from now?
Yeah, one of my favorite ways to also frame it differently that gets people really excited is to think about their pets, right? Pets might live, let's talk about, on average, let's just say 10 years, right?
Uh,
Well, there are interventions now, like even rapamycin, which have been shown to increase dog lifespan by 30%, 33% to be exact. That's another 3 years of life for a pet. And sometimes it's hard for people to say, oh, I'm worried about what's going to happen 45 years from now. But if you put it into that context, right, of, oh, they're my family members would want me to be better during that time, or like all the same context of a pet, it makes a lot of difference, right? You can see that there are other people depending on you, that there's a lot of time left, and that time means something to both you and other people. And so yeah, I would encourage people to tackle it, but also to feel reassured now that aging is no longer it's not Ponce de Leon looking for the fountain of youth, right? It's a science driven plan with clear interventions and ways to manage. And we don't have ways to double our lifespan just yet. They might be coming, but we do have ways to make incremental change, which will absolutely play a difference in your life.
it's awesome. People hear about Brian Johnson a lot.
Yeah.
He pushes he's great for us because he's bringing such awareness to this field. And they always have these competitions of who's aging the slowest. All right.
Yeah.
Let's talk into some of the validity of some of this, and
Yeah.
which tests really are showing that, and how accurate is the Dundon pace test?
Yeah. Yeah, so this is an important point because with so many different clocks out there, it's important for people to know how we judge good clocks, right? What exactly, how do we know that one is better than the other? How do we know when to use one versus another? And this is extremely important because we do this based on outcome data, right? So in order to see how good a clock is, we have to take samples that were taken 30 or 40 years ago. And then we have to see which clock is best at predicting outcomes over the course of a long time, right? Because that's essentially what we're wanting to do, is to say, can we create a predictor for outcomes? And if that predictor is really accurate and we change that predictor, can we change the outcomes? And the answer is yes, absolutely. So we judge the efficacy of these clocks. So whenever we're asking about how accurate a clock is, generally we're saying how good is it at predicting outcomes, right? And also the secondary thing we're asking about is how reproducible is it? So if we take one sample and we test it multiple times, how much variation is there? Because if there's 5 years of variation, then if unless we see a 5 year delta, right, in your age, we're not sure if that actually is meaningful. And so we measure these by hazard ratios, how predictive they are of outcomes, and then also interclass correlation. So if we take the same sample multiple times and test it, how close to each other are each of those results. And so the gene pace is actually performs exceptionally well on all of those testing. It's one of the most predictive of outcomes. It is certainly the most predictive of comorbidities. So more of that health span lifespan related mark. And it's also, it only has right around, I would say, a 4% possible technical error. And now that's even generally around 2% in most of the studies. So if we're taking a sample, generally it's not going to, and we test that sample multiple times, it won't vary more than right around 2%. So that's what we usually would say whenever we're talking about the accuracy. And so people like Brian and we've certainly created the Rejuvenation Olympics as this competitive leaderboard where we've chosen the Dunedin pace, where we have a couple different rules on the leaderboard, like you have to take the test at least 3 times to make sure that variation or that technical variation is very clear. But Brian in particular started with a rate of aging of 1.01 the first time he ever took our test, which, to be honest with you, is not very good. It's we say if you're over 1 or you're aging faster biologically and chronologically, that's a really big risk. You're at a 56% increased risk of death over the next 7 years, and you're at a 54% increased risk of a chronic disease diagnosis. So being over 1 is not good. But since then, Brian has done some incredible things on that marker. I think his most recent one was 0.49, which is the lowest I've ever seen. It's pretty incredible. I think he just published that last week. And so, he is, with his data-driven approach, being pretty successful on the epigenetic biomarkers, especially. He originally had a 0.61, which we thought was phenomenal. We didn't even think we could see a 0.45, but now we've again tested a couple times and he's done very well. But he's doing everything and testing very, very frequently as well to see what actually works. And so one of the other things that in that interventional trial we talked about with Yale, out of the top 7 interventions, 6 of those were in populations that were less healthy. And so the big takeaway there is it's easier to change health if you're already doing a little bit worse than average. And that makes sense probably, right? It's easy to optimize something that's not very optimized. And it's harder to optimize something that's already really well put together. So I think that that's an important category. But of those six of the 7, the 7th intervention, which was not in an unhealthy population, was hyperbaric oxygen. And that was one of the things I think that we're seeing be pretty effective, surprisingly effective in helping improve aging. And one of the things that he's recently implemented. So it might be one of the reasons that his most recent pace of aging is so good.
Wow, that's great. So we got hyperbaric oxygen, right? There's pharmaceuticals. From what you guys have looked at, can you go through your list of 7?
Yeah, certainly. So I can say that we didn't, all of the data that I've seen are not all the data that made it into the paper. So I might go a little off script here and talk about some of the things that really excite me as well.
A scattergun approach too. So
yeah, so generally, one of the things we've already talked about at least a little bit is senolytics, and senolytics are really interesting. If you look at the main paper or the main figure from that trial, one that a lot of people tweeted out and talked about, it's not the best figure to actually understand the results. And the reason being is that we used every possible clock, not just the good ones, right? We've been talking about the good ones on this conversation, but we used even those first generation clocks developed over a decade ago, and so they just give a little bit of misleading data. So for instance, on the first generation clocks, we see biological age go up with senolytics, and we would say that that's probably not the right direction, especially because in the newer generation clocks, senolytics like dasatinib and quercetin actually improve biological aging. And so this speaks to why the generations of clocks and newer clocks are so important, is because we're getting data that makes sense. One of the other examples here is caloric restriction. The calorie study was a 2-year trial on right around 11% caloric restriction. So eating basically 11% less calories than you're expending every single day. And this is another good example, is that most of the first generation clocks didn't respond to that. The 2 clocks that went in the right direction were Grimage and Dunedin pace. And Dunedin pace was the only one which was significant, which is why it became the leaderboard standard for the Rejuvenation Olympics. And so, but again, if you combine all those clocks together, it might look like you're not getting that big of an effect size. But if you look at the clocks that really are the most important, we see that, again, they're behaving exactly like we would expect. And so caloric restriction, hyperbaric, exercise, and then even senolytics all actually take those down.
That's awesome. And, it just goes along with what we're preaching here, and that we're giving information to people. One of the things that I find a little crazy in a lot of the main podcasts for health is how much they're pushing high protein diets.
Mhm.
It just doesn't work when you're looking at these clocks because
Yeah.
when you start pushing these amino acids that drive growth, that drive mTOR, you're aging fast. You know,
Yeah.
and maybe you can, I can go on a little bit, but I'd
Yeah.
hear your input on what you're seeing with the people on high protein diets.
Well, first and foremost, in most of the data sets we looked at there, we didn't have a lot of high protein, but we had things like vegan versus omnivore. We had Mediterranean diets. We had, some of these things. And generally, I won't bury the lead. Out of the data that we had in that published trial, Mediterranean diets do phenomenally well. These
Right.
anti-inflammatory diets with high polyphenols, healthy fats and oils, they work well, right? The Mediterranean sort of Greek diet is excellent across the biological age clocks. But when we talk about the other protein type of things, we get into other known longevity interventions, which tend to be pretty good. So things like methionine restriction, right? Methionine is found really high in eggs. That's a longevity sort of benefit or protocol because these small branch chain amino acids tend to be proliferative. And it brings up, another question here. I don't know if we want to get into it at the time, but like even for instance, growth hormone stimulation, generally higher growth hormone levels, higher IGF-1 tends to be not as good for overall lifespan. And so, we're always trying to balance, some of the quality of life with overall lifespan, which is why these definitions of aging are so important.
Yeah, and I'm also finding like the timing of these epigenetic tests matter a lot, like with the interventions that we're making.
Mhm.
Like I try not to get any epigenetic clocks right after we do a dasatinib cycle, right? Because
Exactly. Mhm.
inflammation, but then it's going to drop afterwards. Um,
Yeah.
and so
And
everything has to be timed at the right time, and that's why we have to have this project management set up. And
yes.
it's funny because doctors don't know engineering. They're problem based and they don't have project management suites. Like the EMRs are not set up this way. That's why we try to use engineering protocols, and it's nothing that I created. It's like maintenance engineering is like 100 years old, right? It's why
Yeah.
planes don't fall out of the sky all the time. I mean, pretty rare. Um,
Yeah.
and why people have to change, they're like, oh, we've changed people's diets around. Like now every quarter they do fasting mimicking diets. We used to do
Mhm.
water fasts, but now we're finding
Mhm.
fasting mimicking tends to beat a water fast.
Mhm.
I'm trying not to get people to do too much intermittent fasting because I find that when they eat in too short of a window, then their bodies start making too much glucose.
Mhm.
I'm trying to get people to not eat 4 hours before bed. It seems to be
We
a great intervention.
Yeah,
Um,
it's funny you mentioned that. We just did a study on Ramadan,
yeah.
where it's basically a time restricted feeding diet because you sort of you overload after the sun goes down and et cetera. We saw exactly the same thing.
Yeah, I mean, there's been some studies where like they give people the same amount of calories and
Mhm.
the same food, the same exact food, except some people get their food early in the day and other
Mhm.
people get their food late at night. And the
Mhm.
people that get their food earlier in the day have better energy and they lose weight or they maintain their weight. And the other people have lower energy and they gain weight. Uh,
Exactly.
and
Mhm.
I like looking at like the monks that have been living in monasteries for thousands of years and the way that they do it. And it seems that like most of these monasteries around the world do kind of the same thing. They wake up early, they eat most of their food early in the day, right? And they have a small dinner and then they fast before they go to bed. Um,
Yeah.
and it's like, all right, that's trial and error over hundreds and hundreds of years. They kind of figured it, right?
Yeah,
And
definitely.
our
Yeah.
science a little bit behind that, but
Yeah.
follow those kind of protocols, you're going to do pretty well.
Yeah, absolutely. Going back to Brian Johnson, the number one thing that he optimizes is sleep. He thinks it's the most important, and to do that, it's stop eating a couple hours before bed. But obviously he's doing a lot of caloric restriction as well, and it's all the things we've talked about, right? Because generally the science keeps pointing back to these definitive options, and again, I think that's super encouraging, and that's why in our study we found a lot of interesting things like the response to dasatinib and quercetin, or the response to hyperbaric, really novel couple, things that we didn't quite see, but we also saw the things we would expect to see, right? Which is caloric restriction being better, or the Mediterranean diets being better. And that's also super encouraging as well, because it backs up what we already know in all of these, decades and decades of information and science.
It's awesome. And we know that rapamycin, there's tons of studies on rapamycin that's working really well.
Mhm.
Here, what we're doing with rapamycin, and I don't know a lot of other people that are doing this, but like we don't give rapamycin every single week to people.
Mhm.
We do a little bit of a break and we allow them to stay off the rapamycin, and then we try to get them to increase their calories just a little bit and go up on their weights, just to add a little bit of muscle mass so that they don't become sarcopenic.
Exactly. Yeah.
Some of these people on long-term rapamycin without breaks become sarcopenic, and I think that's going to affect their aging in a detrimental way. Uh,
Mhm.
so we have some of these protocols, and it's been working really well. Our testing is coming out great for that. Um,
Mhm.
what
Yeah.
I'm interested in now, as we're wrapping down to the end of this podcast, is what's the future that TruDiagnostics is working in? Like, what are some of the things that you think that you're going to be able to provide in the future, and what's the future of
Yeah.
Universal in your
Oh, so that's a hard question to answer. And first, I think I want to answer it maybe two ways. One is on, what I think is most exciting about the testing, and then on the other side, what I think is most exciting in the interventional space. And so on the testing side of things, I really do think that DNA methylation can be one test which captures a variety of important information, all in one test. So right now, again, we can tell you about your omega-3s, your VLDL particle sizes, your NAD pathways, all with the same drop of blood, that we can tell you about your biological aging or your immune cell subsets, or all of these different things. And very soon we're going to be adding another one of that, which would be what we call methylation risk scores, which is very similar to the disease predictions you get from a 23andMe or some of those other things. But the good thing about that is it's also changeable. So then we can go back to those same interventions and say, what actually works to reverse cardiovascular disease risk, or diabetes, or COPD, and all of these other things. And so I really do think that just generally, conversations about aging also have to start to include disease, because we know it's the biggest risk factor for all disease. And if we can get that all together in one test, I think that's so valuable to be able to predict outcomes and steer us in the right direction. So I think that's what really excites me is a platform where one at-home blood spot test can be able to tell you an infinite amount about your health. And that's why I'm super excited. So we can start to consolidate all of this and not just compartmentalize it, right? Not just compartmentalize, the 9 hallmarks or 11 hallmarks or 15 hallmarks of aging, or your blood-based biomarkers, or, all these things, but include it into a single model that's really, really informative, really inexpensive, and able to do at home, right? I think that that's what excites me about the testing, and we'll start to be able to see what's great for aging, what's great for disease, where they conflict, and start to answer some of those hard questions. But on the interventional side, I mean, I'd be remiss if I didn't talk about cellular reprogramming. And I know that you know about this. I'll try and intro it for anyone who doesn't. But in 2012, there's a Dr. Yamanaka who won the Nobel Prize. And what he was able to do is to take certain cells in your body and reset them to pluripotent stem cells. So sort of do a reset. And in recent years, what we found is that reset, those growth factors which can reset the epigenetics, actually take those clocks back to zero, right? So if we had if we, for instance, take a sample of your blood and split it into two, we run a test on your blood, and we get your age. But then on the other sample, we use these Yamanaka factors to reset your epigenetics. That clock then goes to zero, or goes down significantly. And I think that this is one of the most exciting things in health, is this sort of epigenetic reset. It's sort of a restart. And how that will actually take place in the clinical setting over the next 10 to 20 years, I don't know. It could be that we start regrowing identical organs, right? So that we just start replacing our hearts or our livers, or all these things, because we can grow it from scratch from a cell, a skin cell that we get, for instance. So and I think there's a lot of really cool applications there. And so that's probably what I'm most excited about interventionally as well.
Yeah, cellular programming is really amazing. You can do Yamanaka factors, and you can also do it with infusions of pluripotent cells. Because pluripotent cells, if they're yours or if they're from a donor, they express Yamanaka factors. And
Mhm.
so not as much data with that, But one of the early problems they thought with Yamanaka factors is like, okay, if we give you Yamanaka factors, we're going to increase your incidence of cancer. But they're actually finding the opposite,
Mhm.
that by giving these Yamanaka factors, by IV, that you're going to decrease your cancer risk. And I think that's a game changer. And it's definitely on the radar for both Pazio Institute and the IMME, of when do we have enough data and when will we have the right protocols to begin using that?
Yeah, absolutely. And I always like to mention Life Biosciences. The company is actually going to clinical trials in the next year to help restore blindness or vision in blind individuals. And so it's not out of the clinical picture, for, a lot of people think it might take decades, and it might to get it in a large scale population, but it's already being tested on humans and to restore, vision, which is an incredible thing. And so I think everyone should get really excited about that. And I think that the fact that it's mediated via this mechanism, which we know is one of the best ways to measure aging, I think is even more exciting because we're drilling down into maybe a process that is incredibly important for our health.
So many good things happening. I don't even know how to wrap all this up. I, Definitely go to the True Age site, everybody, just so you can read about some of this stuff. It's trudiagnostic.com, TRU diagnostic.com. And you can learn about the tests. You can even get your own test. Obviously, if you come to the Pazio Institute, there's going to be one of many tests that we do in what we call the discovery process. Discovery process, if you just Google discovery process in relation to maintenance engineering, you'll see that what engineers do is when they come to a complex system, they have to figure out what's going on with that system. What are the single point failures that are most likely going to happen in this system state? And so this is one of the pieces. But, for those of you that are not part of the Institute, definitely go do this because you can help yourself tremendously just by knowing what's going on with you. How are you aging? Right? Where's your inflammation at? Look it through all these tests and change your lifestyle for this. Get a health coach, do what you need to do, improve your sleep, follow some of these podcasts. You follow our podcast. And I think that this is a game changer. And these tests are just getting better and better. I mean, the level that you guys have gotten to and the amount of markers that you're looking at is mind-boggling. And without the machine learning and the AI of today, it wouldn't even be possible to use the information.
Exactly. Even 2 years ago, we couldn't create the models we're creating now, it's incredible.
It's awesome. Well, thank you so much for being on the podcast, Ryan.
Yeah.
Pleasure, and we learned a ton. And looking forward to the future and the
Yeah.
next steps that you guys are coming up with, and the interventions, and showing us what's really working, and collaborating with us in all the different ways.
Likewise, yeah, we pride ourselves on the close relationship with physicians because you all are out there actually doing the information that helps us learn, and so hopefully we'll be on again to talk about our next major breakthrough.
Oh, fantastic. All right. Nice seeing
Thanks
you.
so much.
Bye bye.